Okay, hello all. I hope you're having a good day. So today we would like to present on our ABC project, which is documentation of Basidiomycota in Santubong National Park, Sarawak. So first and foremost, I would like to introduce our group members. So the first group member would be Muhammad Faiz Suhaili. He is our group leader. Next, Nur Hazia Amni, Eliana Winnie, Laurentia Guyang, Nur Afika, Umi Husna, Nur Aizira, Yugendran Mahendran, Amira, Kasdina, and last but not least, me being the representative to present our documentation, Jason Lim. So here is our overall poster. No worries, we will break down one by one so that we will have further clarification. So here is our abstract. We would like to document on Basidiomycota, which was conducted in Santubong National Park. It's known for its tropical forest with abundant fungus to study on the external features and characteristics of different types of Basidiomycota. So this study was done through observation methods and identified many Basidiomycota species in Santubong National Park. However, we focus on 3 species with 3 different characteristics to show diversity of Basidiomycota fungi. Next, the introduction. So Basidiomycota is from the monophyletic group with more than 31,000 living species and are known, but molecular and genetic studies indicate that there are more varieties that are yet to be studied. So in our opinion, the importance of carrying out this documentation is to understand the role of Basidiomycota in the ecosystem, as Basidiomycota are known to be the main degrader of different components, as they are known to be saprophytic. So this documentation focuses on 3 species and is done through the process of jungle tracking through the loop trail. So the methodology used was through taking pictures using the smartphone. So once the pictures are taken, they are compiled, sorted, and recorded. After the trip, the pictures were examined, and whichever picture that suits for this documentation would be selected. Not forgetting, we did use a 30 centimeter measuring ruler and a 10-cent coin to measure the size of the Basidiomycota species. So these are the results and discussion. As mentioned just now, we actually found a lot of Basidiomycota. However, we focus on these 3 species. The first species discovered was Polyporaceae sanguineus. Forgive me for my incorrect pronunciation. So morphologically, it is usually seen as a bright reddish-orange fungi and is known to be saprophytic. So based on the picture, it grows on dead wood. It is known to cause white rot. Next would be the Ganoderma aplanatum, or also known as bracket fungi. In terms of appearance, it is usually dull brown in color. So this species is known to be both parasitic and saprophytic. However, for this specimen, during the loop trail, we found that it was attached to living trees. Hence, we believe that this specimen was parasitic. Last but not least, we found the species called Microsporus centophus. Morphologically, it is brown in color. So this species is saprophytic. As can be seen clearly, it is found on rotting wood and usually seen in tropical areas. So as a conclusion, throughout the loop trail at Santubong National Park, we found 3 Basidiomycota species. We observed it and recorded, which is Psychosporus sanguineus, Ganoderma aplanatum, and Microsporus centophus. All 3 species were found in around the distance of 1,500 meters to 2,000 meters along the loop trail. So as for acknowledgement, we as the authors would like to thank the Center for Pre-University Studies, University Malaysia Sarawak, who provided us the financial support and also transportation. We would also like to thank the Sarawak Forestry for the visiting permit for the success of this study. And last but not least, we would like to thank the Biology Unit Laboratory staff as well as the lecturers who assist us throughout the studies. And not forgetting, we would also like to thank our fellow members who spent their time and effort in creating the success of this study. Thank you. Here are the references.